And our first speaker is Miriam Merad, which I happen to know since 18 years, as we just recalculate back together. So Miriam is a professor of oncological science at the ITCAM School of Medicine at Mount Sinai, so again, not too far away from Rockefeller. She's as well the director of the Precision Immunology Institute, and she has really done absolutely fantastic, groundbreaking work on understanding myeloid cell biology, and she will tell us more today about what she has discovered about the tumor microenvironment. So Miriam, thanks for coming.
Okay, so thank you, Emmanuel. It's a pleasure to be here. Thank you, Theodore, because in fact we've been trying to meet since she moved from UCSF almost like a few months ago, and now we met again. So it's a pleasure to be here, as always. I thought that my students will attend my talk, but he didn't. Okay, well, I'm disappointed. So today I'll be talking about the tumor myeloid microenvironment, and maybe I can. And among myeloid cells, I'm going to focus on the mononuclear phagocyte compartment. This is a little bit too loud, no? Are you hearing me well? Yeah. And I'm going to focus on this group of cells because these cells are, because of their key role in anti-tumor immunity. So these mononuclear phagocytes are also called antigen presenting cells, and they are cells that are able to really sense the environment. They are injury sensors that can sense tumor growth, they can sense tumor cell damage, they can sense cell death, they can sense microbial signals. And when they sense damage, they release inflammatory molecules, which is also called, this innate immune response that contribute to shaping the tumor microenvironment. So they play a key role in really deciding what immune cells are going to really surround the tumor cells. But they also play a key role in shaping tumor T-cell specific immunity. T-cells do not recognize native antigens and need to be instructed by APC to kill. And we have shown that the checkpoint blockade response, for example, depends on my students arrived. So thank you for coming to attend my talk. And we had shown that tumor response to checkpoint blockade really depends on APC quality at the tumor site. So the tumor-associated antigen presenting cells include 3 cellular compartments that consist of monocytes that are present mostly in the tissue, that can also accumulate at the tumor site, dendritic cells that you can find in the blood and at the tissue site, and macrophages. And tumor macrophages are often the largest immune cell compartment in tumor lesions. They have been shown to play a key role in tumor immunity through tissue remodeling and modulation of tumor immunity, yet targeting macrophages in cancer has proven to be very difficult. And we believe that this is due to the fact that knowledge of human macrophages is mostly based on monocyte-derived macrophages that have been generated in vitro in defined culture system, which in fact led to this M1, M2 paradigms where all macrophages are equal, but responses is dictated by external cues. And this has been a very useful definition, but it remains an incomplete definition, which, as it ignores an important compartment about these macrophages, which is called the tissue-resident macrophage lineage. So my laboratory have used fate mapping studies of both the embryo and adult hematopoietic precursors to show that, in fact, many macrophages in tissue are, in fact, recruited to the tissue prior to birth and derived from early embryonic precursors. In the brain, for example, macrophages arise from these yolk sac EMP cells that are recruited to the brain a few days after conception. We had shown that most macrophages in tissue arise from this second wave of myeloid seeding that occurs in the fetal liver. And then you end up with a pool of macrophages in tissue that is maintained in the animal for a very prolonged period of time. And consistent with this life cycle, we had shown that macrophage, in fact, in tissue, have a unique transcriptional program that is really dictated by the tissue in which they reside. You will have a very different picture if you were, for example, mapping transcriptionally dendritic cells in tissue, which have much more in common than these macrophages. And we had shown also that epigenetically, these macrophages have a very distinct enhancer landscape that's also dictated by the tissue in which they reside. So and what we found is that, in fact, in most tissue, at least in the steady state, most tissue are inhabited by this pool of macrophages that are recruited very early during embryonic development. These tissue are also inhabited by another large APC, well, not large, but important APC compartment called dendritic cells, which we often refer to as bona fide dendritic cells. These dendritic cells, in contrast to the tissue-resident macrophage that I've just described, arise from dedicated precursors in the bone marrow that are constantly recruited to the tissue to maintain the cellular compartment. However, in the inflamed setting, and for example in lung tumor, which is going to be really the focus of my talk today, there is also an influx of monocytes from the bone marrow. These monocytes that are recruited to the site of injury can differentiate into cells that can resemble these embryonic macrophages up to a certain point, and I'm going to get back to this extensively during my talk, and then can also differentiate into dendritic cells that resemble the bona fide dendritic cells. But despite the fact that they share some program, we believe that these compartments are quite different. And understanding the differences of this compartment in their ability to instruct T-cell immunity is probably going to really help identify the role of these different myeloid cell compartment in tumor immunity and potentially, in fact, identify relevant myeloid-specific targets. So very little is known, indeed, about the extent of APC diversity in human tumors and their contribution to tumor immunity or tumor progressions, and hence the need, we believe, for a better understanding of myeloid composition in human tumors. Now, because cells are really the functional units of biological processes, we believe that mapping cells, in addition to pathways, can provide critical information on both the disease pathophysiological processes, but also on target prioritization, which I'm going to discuss later during this talk. So my lab focuses on early non-small cell lung cancer because of the high incidence of lung cancer-related death, because lung cancer have a significant response to immunotherapy, at least for advanced lung cancer lesions. These early lung cancer lesions have been shown, in fact, to have an increased response rates to checkpoint blockades, has our interest in really studying immune defects that occur in these early lesions. But also Sinai has a very large access to surgical resection due to a very strong lung screening program that provides access to more than 400 early lung cancer lesions per year. So to really have a first pass of understanding of both the myeloid, but also immune changes that occur in these early lung cancer lesions, what we did is we made use of a mass cytometry platform, and that we used to, in fact, map the immune cell compartment in initially early stage lesions, and we recently extended it to later stage lesions. And here what we did is we collected the tumor, the adjacent tissue, and the blood of the same patients. Each tissue was barcoded with a metal. The sweet tissue were pulled and then stained with two extensive lymphoid and myeloid panels. And we found, in fact, very discrete immune changes already in stage 1A lesions. And we showed, for example, there was a very significant accumulation of T regulatory cells, of PD-1 positive T cells, and in these lesions compared to the adjacent tissue already very early during tumor progression. We saw also there was a very strong depletion of NK cells, and there were many changes in the phenotype of myeloid cells. But what we also realized is that there were many cells expressing canonical myeloid markers that we couldn't classify as a discrete myeloid population. And this is because the antibody panel that we are using are really based on prior knowledge. Right? So what we decided to do is start to develop an unbiased mapping approach by which these tumor lesions will be now analyzed using single-cell RNA sequencing, which in fact can be done regardless of preconceived knowledge. And using the same strategy that we use for mass cytometry, we collected the tumor, the adjacent tissue. We digested the two compartments. The two compartments were barcoded, first at the cell level and the tissue level, and then encapsulated with B that were coated with also barcoded primers. And then we encapsulated the cells using this 10x platform, and then we sequenced, right, every cell from both the tumor and the adjacent tissue. So we have now analyzed 29 patients with adenocarcinoma lesions. We sequenced 500,000 immune cells. And this is a summary of some of the results that we obtained. In these figures, the row represent one gene. Each column represents a cell. Each color represents a patient. And then blue here refers to the adjacent lung. The brown refers to the tumor. And then, so here I'm showing you only the pictures. I'm just going back now to the macrophage compartment. I'm just showing you our dive analysis on the diversity of macrophage population. And after something like 18 months of analysis, we now have, I think, a good idea of what happened in these tumor lesions. So first, the surprising thing is that many of the clusters that we identify, in fact, are very well distributed among patients. So we didn't identify yet a very discrete, at least with the higher view, distinct group of patients. So there is a very nice distribution of clusters among patients, which is quite reassuring when you think about targeted therapy. And then we started to look at what clusters were enriched in the tumor versus the adjacent tissue, and we identified clusters that were strongly reduced and clusters that were, in fact, increased. And we are now trying to really focus on those clusters that are strongly induced at the tumor lesions and those that are also reduced at the tumor lesions. So this really helps you start to reduce myeloid cell complexity. You can start to look at your favorite family of molecules. So we are very interested in cell surface receptors, and you can now start to look at all cell surface receptors and ask how they are distributed among the clusters that you identify. You can also focus on one family of receptors, as we are doing, and then look at their distribution among clusters, and then decide whether you want to explore those that are specifically expressed on the cluster of your choice, or study those that are equally distributed among clusters. And what we are also doing is looking at how these clusters are, in fact, changing in patients that resist or respond to checkpoint therapy. And so we designed, together with the Regeneron group, a series of clinical trials where patients with lung adenocarcinoma will receive, prior to surgery, 2 doses of cemiplimab, which is Regeneron PD-1 blockade, another one. And patients with resectable HCC will also receive 2 doses of PD-1 blockade. And we will isolate now the tissue prior and after these 2 doses of checkpoint blockade and ask how these clusters that we identify are going to, in fact, changes. Are they enriched in patients that are resistant, or are they, in fact, reduced in patients that respond? Okay, but what we also wanted to do is really start to probe the ontogeny of the myeloid compartment in these tumor lesions. Now, we cannot fate map an animal, so a human. So what we decided to do is to go back to the mouse and try to see whether in this tumor lesion, these tissue-resident macrophages were, in fact, persisting in the tumor and whether they were, in fact, contributing to tumor progression. So here we used two different tumor models of lung adenocarcinoma that were developed by Tyler Jacks. In these tumors, these lung epithelial cells express a KRAS mutation and a p53 flux allele, and we use either a tumor graft or we directly induce tumor development using this adenovirus CRE that Tyler Jacks described. So we obtain very nice lung adenocarcinoma lesions that resemble the one that we see in patients. We have now sequenced the myeloid cell compartment in both tumor models. And this work was done by Maria Sebes, who is in fact in the audience. And what Maria found is that the myeloid cell compartment in these two tumor models is quite similar, so that's great. And now she's going to ask whether some of these, so we find again these two clusters of monocyte. Maybe I didn't mention it, but in human we find two clusters of monocyte that really are in line with what's published in the literature, which is always reassuring when you see that. It's quite crisp. We also find two clusters of monocyte, the classical and the patronic monocyte, in these mass tumor lesions. And then we find three big clusters of macrophages. And now she's going to ask whether among these macrophage clusters, there's any that are coming from the embryo or whether they are all recruited from adult hematopoietic stem cells. So to do that, we used a model that was described by Boris Reisis at NYU. And Boris used this molecule called MAP17, which function is not well understood. MAP17 is expressed in early hematopoietic stem cells. And he developed this, and he showed using this MAP17 CRIR crossed with ROSA20C tomato, he was able to fate map both the hematopoietic stem cell and its progeny. So upon TAM injection, he showed that HSC was highly labeled in these mice, and he was also able to label circulating lymphocyte, circulating granulocyte, and megakaryocyte. However, he was unable to label macrophage in the tissue, which is consistent with our previous finding, showing that macrophage in tissue, in fact, do not derive from adult hematopoietic stem cells, but derive from these embryonic precursors that are recruited to the tissue prior to birth. So we were unable to, in fact, label this macrophage compartment, and these data have been reproduced by many groups. He also showed that, in fact, in mice that were inflamed upon viral, I think, infection or injection of Toll ligand, he increased the labeling of circulating lymphocyte and granulocyte, but was unable to label the tissue-resident macrophage. And this is what we are showing here. So here in a normal lung, we see that we, in these mice, obtain very nice labeling of circulating monocyte and circulating neutrophil, but we didn't, and we also labeled monocyte in the lung and neutrophil in the lung, but we didn't label tissue-resident macrophages. So this is in a normal animal, recapitulating, in fact, prior data from my groups and others. So now we are going to do exactly the same experiment in the tumor-bearing animals using the same fate mapping model. We are going to first inject tamoxifen into this MAP17 CRIR mice and wait 6 months first until we obtain sufficient labeling of circulating cells, and then inject the tumor lung adenocarcinoma cells. And 4 weeks later, we purified red cells. So these cells are derived from adult hematopoietic stem cells, or we purified non-labeled cells, suggesting that they don't derive from hematopoietic stem cells, or there was some labeling deficiencies. And we sequence again at the single cell level both the red cells and the non-labeled cells. Okay? And now we are going to ask where these clusters are enriched, the cluster that we identify at the single cell level, whether they're enriched in the red compartment or the non-labeled compartment. And if they are enriched in the non-labeled compartment, it will suggest that they don't derive from adult hematopoietic stem cells. And this is what we find. This is really a brave experiment. You can see how long it takes to obtain these results. And I'd like to thank, in fact, Maria for her patience and passion. And what she found, in fact, quite beautifully, is that one of the clusters that she identified in the tumor-associated myeloid compartment was, in fact, never enriched in this red cell, in this red cell compartment. In contrast, most of the other, both the monocyte compartment and other macrophage compartment, were strongly enriched or were at least contained some red cells. So this suggests that this compartment here, these clusters here that we identify in the lung tumor, do not arise from adult hematopoietic stem cell compartment. So it's emphasized that even in an inflamed environment, these embryonic tissue-resident macrophages continue to form a transcriptionally distinct macrophage compartment, suggesting that it may contribute, in fact, to tumor outcome. Okay? So this also allows us now to identify novel markers that help distinguish the ontogeny of this macrophage compartment in the tumor lesions. And we found, using the single-cell analysis, that this cluster 1, which is the embryonic macrophage compartment, express some specific marker, including MRC1 and Siglec1. So we made a reporter mice, and using this reporter mice, we are able now to see that we enrich, we label mostly this embryonic macrophage compartment. So now you don't have to do single-cell analysis anymore. You can just use a detailed phenotypic panel that has been informed by your single-cell analysis. And you can also now start to assess spatial cell contextualization. You can go back and see where these embryonic macrophages are localized in the tumor microenvironment. And what we found is that these embryonic macrophages, in fact, accumulate around the tumor lesions, which was quite surprising to me because this embryonic macrophage should precede tumor development. And we thought that they will be, in fact, completely intermingled in the tumor lesions. So what Maria decided to do now is to start to look at macrophage-tumor interaction at different times during tumor development. And what she found is that, in fact, macrophage, this embryonic macrophage, now that she labels using the specific markers that we identify in the single-cell-seq approach, so she found that they are the first to interact with tumor cells. And what's striking is that, in fact, they are always in excess of tumor cells. And then during time, these macrophages are going to be redistributed around the tumor lesions, as if they were shielding, in fact, the tumor lesions from the external environment. So that was very striking to us, and in fact reminded us of a study that we published last year in collaboration with our colleagues Julio Aguirre-Guisso at Sinai, where we showed that embryonic macrophages, in fact, are the first to be recruited at the tumor site. Here we were studying DCIS, so breast cancer in situ lesions, and we showed that this early recruitment promoted early dissemination of cancer cells. And this early dissemination was due potentially to the induction of this epithelial mesenchymal transition phenotype. So it promoted, in fact, the induction of a mesenchymal phenotype that allowed the cells to escape. So we decided to assess whether these embryonic macrophages were, in fact, promoting tumor cell invasiveness. So what we did here, we used this spheroid model in which we will co-culture lung cancer cells with either embryonic or adult macrophages, and then we assess whether the addition of embryonic macrophage will, in fact, promote tumor invasiveness in this matrigel spheroid system. And strikingly, we find that indeed the addition of these embryonic macrophages that were being purified from the lung did induce this, what's called this bridging colony. And we think that these bridges, in fact, reflect tumor invasiveness, reflects tumor ability to, in fact, get rid of this matrigel. But when we added adult monocyte here, what we found, we never detected this bridging colony. So they seem to have a very distinct role here. And this is also in line with this cell dynamic that we observed. These embryonic macrophages indeed are the first to be recruited and potentially contribute to tumor growth by promoting tumor invasiveness. But we were also, and we then looked whether these embryonic macrophage were also able to promote this EMT phenotype. And indeed, we found that addition to the addition of alveolar macrophage, again in the same system, strongly reduced the expression of ficadherin, which is an epithelial marker, and potentially contributing to this EMT transition. You reduce your epithelial marker and start expressing mesenchymal marker, which allows you indeed to be much more motile. All right, so I'm an immunologist, and so I'm going to go back and look at what this means immunologically in the tumor microenvironment. So what we saw during, when we were studying, in fact, tumor evolution, we also saw that along with this redistribution of macrophages around the tumor lesions, we saw that T cells were also redistributing around the tumor. And while these T cells were redistributing around the tumor, we saw an expansion of T regulatory cells during tumor growth. So that was quite interesting to us because we had the opportunity, in fact, to study very early lung cancer lesions, thanks to our strong lung cancer screen at Sinai, where we have access to these lesions are recognized by radiologists as being in situ lesions. They are called ground glass lung cancer cells or part solid nodule. And these part solid or ground glass nodules are usually shown to be histologically non-invasive adenocarcinoma lesions. So we studied the immune compartment of these non-invasive lesions vis-à-vis these invasive lung adenocarcinoma lesions. And what we found is that during this transition from non-invasive to invasive, we saw a sharp increase of the T-reg compartment, which was associated with the reduction of CD8 effector cells. So, which is what we saw, in fact, what we saw also in our mice. So what we decided to do is to ask whether this change of T-cell compartment could be, in fact, instructed by this embryonic macrophage compartment. And because now we know how to really analyze the cells or identify this embryonic macrophage, we developed a deleter mice that will delete specifically the embryonic macrophage compartment. And what we saw is that when we depleted the CD169 compartment, without affecting the adult macrophage compartment, we strongly, in fact, delayed the tumor growth. And we also reduced T-reg accumulation and increased CD8 T cell accumulation in tumor lesion, as you can see here. So, and locally, in fact, the result was much more dramatic. So usually we take the whole lung tissue and analyze it in bulk by flow cytometry. But when we look at really spatial distribution of T cells, what we saw is that when we deleted this embryonic macrophage compartment, we saw an accumulation of T cells in the tumor lesions. Okay, so then we went back now to our human single cell analysis, and we tried to now identify a macrophage cluster that resemble the embryonic macrophage clusters. And of course, we are doing this for all macrophage clusters, for which we are going to probe its function in mice. What you can do is go back and try to do this cross analysis to now infer different function to each of these different clusters that we identified in the human, using, in fact, the mouse as a functional system. So we believe that we identify clusters in a human lesion that we think resemble very much the mouse clusters. So how do we go from now? So what we wanted to do now is to identify, in fact, a phenotype that will allow us to purify macrophage from this tumor lesion. So for this, we are now using this new technology that we love in the lab, the CytSeq, which will integrate both cell transcriptomics and proteomics. So using the CytSeq technology, what we can do is we can now label the cells with antibodies that have been conjugated with an oligonucleotide, which will serve as a barcode. And then the cells that have been labeled will then be encapsulated the same way that we've done by single-cell RNA sequencing. So, and we, again, use the 10x platform now to analyze both the antibody and the transcriptomics of these cells. And here, the antibody library and the cDNA library have different size, so they will be, in fact, sequenced separately, isolated based on their size and sequenced separately. And I have to say this is working quite well. So we were able to now go back to our non-small cell lung cancer lesions, and we now analyze also the entire immune cell compartment using both transcriptomics and the 47 antibody panel. So we can now gate on these clusters that we believe is quite similar to the embryonic macrophage cluster that we identify in mice, and then really assess its phenotype. So, for example, here, this cluster that we think refer to this tissue-resident macrophage, express CD11C, express CD206, doesn't express CD38. This will allow us now to purify these clusters and potentially now assess its role in tumor growth, assess its role on T-cell immunity, and we'll be using the same spheroid model that I've described earlier to really now probe the role of these different myeloid cell contribution. We will go, of course, beyond the embryonic macrophage, and you can look at dendritic cells compartment and whatever population you are interested in studying. So with this, I'm going to conclude by saying that there is that different temporal and spatial waves of ontogenically distinct macrophage accumulate in tumor lesions. Adult hemato. Am I very late? Sorry. He has much more interesting things to say than me because he's going to talk about patients. So maybe I should just skip this. I'm thanking my lab, and I'm going to stop here and address any question you may have. You need to tell them.
Regarding, in stage 1 small cell lung cancer, you showed that most of the macrophages are tissue resident. What about cancer cells that metastasize from breast, prostate, and other type of cancer? Are also tissue resident macrophages associated with newly arriving metastases, and gradually as the lesions become larger, are they replaced by bone marrow-derived macrophages?
I'm not sure I completely understood, but yes, the adult macrophage arrive late in the tumor, absolutely, and they accumulate, and they form, in fact, the largest compartment. I don't know whether you pay attention, but at some point I was mentioning this cluster that are reduced in the tumor lesions. So the embryonic macrophage, we believe, are excluded from the tumor lesion, and they protect them. They probably protect them from cytotoxic immunity, but there is accumulation of adult hematopoiesis macrophage. Is this your question?
What I mean is the findings are in small cell lung cancer, so the tumors originate in the lung. I mean, yes. What about metastasis? Cancer cells metastasize to the lung.
In metastasis.
Yes.
Well, I don't know. I don't know whether similar things happen in metastasis. Now, you know, these tumors are sometimes injected IV. When they are injected IV, you can imagine that they are recruited from the blood vessel and may mimic metastasis. So I'm guessing that probably similar pattern will occur in metastasis.
I will take a question. So. Yeah. What excludes this macrophage from the tumor?
Well, so I think, so we believe that they are excluded because they are going to help build a stromal capsule. So our hypothesis right now is that, in fact, what they do is they go activate fibroblast and really encapsulate, help encapsulate the tumor lesions.
What drives this signaling? So
what drives that? Typical referee question. I don't know yet. How? Potentially, yes, but we didn't do the experiment.
You don't have signature in your...
Oh, no, we have tons of signature. Yeah. So TGF-beta is expressed indeed, but we have not done this type of molecular dissection yet. But TGF-beta is on top of our list.
Second question. So you talk about the embryonic macrophage, but you still have other clusters which are bone marrow derived myeloid cell, and you have the way to mark them with a MAP17. So are these macrophages more intermingled with the cancer cell?
So indeed they are. They are closer to the lesion. They have a different transcriptional program. I've showed you this. They are probably playing a more important role in in immune response. So they have more immune genes than these macrophages that seems to be more, potentially more, playing a bigger role in fibrosis, although we are still dissecting that. So yes, they are closer to the tumor cells, which is a little bit bizarre. So we need to now track them a little bit better, and why they are closer is something we are trying, in fact, to explore with Christine Moussion at Genentech.
That was true.
Thank you.
My voice is very soft. Okay. If you give chemical, can you deplete these resident macrophages? If you give the, this crude tool we use, clodronate, can you deplete the resident ones so that you could actually see what their dynamics is normally? Or
so this is what we did. So we depleted them.
And so they do die if you feed them toxic payloads, the alveolar ones. So you could actually see how dynamic they are.
Yeah.
Okay, that's interesting. So my science question is, since you show that they promote EMT and maybe they promote T-regs, do they somehow stimulate TGF-beta signaling? Because that would be the kind of reductionist notion.
So they produce TGF-beta and they respond to TGF-beta.
But do they provide it to the cancer cell, which would promote EMT, and it would
so
also
is
promote...
TGF-beta required? I don't know, but you can assume that they may use TGF-beta to promote EMT.
They could have a MMP, for example, that processes it.
So they produce tons of MMP, a lot of TGF-beta. They're not the only one that produce TGF-beta. So macrophage, dendritic cell that we find also, but they produce a good amount, I would say. I don't know how to quantify, and a lot of MMP. So they are very well wired to do that.